Hello everyone, nice to see you all. Today I will be talking about our work on diffusion-based generative model for gene expression sampling. So I'll start with a brief motivation, then discuss data generation approaches, talk about the usage of AI accelerator, and show the current results. Uh, in our lab, one of the large directions of the work is dedicated to anti-cancer drug response studies. Here we want to identify the drugs that efficiently suppress tumor growth. For this, we need to make sure that the cancerous cells are killed. The way to experimentally validate this is through the cell viability assays. The cells that are surviving, left intact, are colored using the membrane impermeable fluorescent proteins, and then cells colored also with the membrane with membrane impermeable proteins, and the healthy intact cells are colored only with membrane permeable proteins. Okay, so we can get those estimates for a specific drug concentration, and once we vary our inputs and making sure that our region of interest for the drug concentration, we fit drug response curve. This model estimates the drug's efficiency for the specific cell line. And this curve can be described using either the threshold at which the drug is 50% effective, or using the integral characteristic such as area under the curve. So as you can see, it's a very cumbersome and expensive experiment, and we would like to get it to the virtual screening purely in silico. For this, we are using the machine learning methods, and the approach is quite standard. We're using the descriptors for the drugs and the cell lines, and training regression models. For the cell lines, one of the most informative modalities is gene expression, and that's exactly what we would like to generate. The motivation behind this is the lack of the data even among the most simple and affordable biological models such as a cell line. As over the years, the scientific community put a lot of efforts in creating open data sets, and the scientists conducted thousands of experiments. But because of the combinatorial nature of the problem, where we have to get the cell line and drug input, the amount of the biological variation still remains quite low, and we have cells only in low thousands, and most of the ones that I showed here are overlapping. In order to estimate the model's robustness and study the regions where the predictions start to become uncertain, we need a large amount of unlabeled data. For this, we are attempting to generate synthetic pseudo cell lines using the generative AI. And with the generative AI, there are multiple methods we can choose from. And there are log likelihood models such as Bayesian networks and energy-based models, but they either require a lot of simplifications to be tractable, or instantly become intractable in case with the large Markov networks, where we need to integrate all states to get the normalization constant. The second approach is generative adversarial networks, and the third one is core-based models, for which the noise and diffusion models is one of the cases. So the generative adversarial networks are built as a combination of two neural network modules, the generator and discriminator. Generator synthesizes new data, and discriminators tries to distinguish it from the ground truth and gives feedback back to generator. One of the advantages of this algorithm is fast sampling, but games are also notoriously difficult to train. There are multiple issues involved, such as vanishing gradients, model collapse, failure to converge, and they require a large amount of data. A better approach, at least for the video and audio generation, was the noise and diffusion probabilistic models. So here we are sequentially corrupting the data to make it indistinguishable from noise. So we are getting the multiple states of the object that is more and more noisy. And we build a Markov chain over those states. The direct process is to add noise according to the schedule, and the reverse process would involve the learning the noise model. It results in a very high quality data samples, but the convergence speed here is quite slow, and the sampling process is also slow because we need to run the noise models for each of the steps. One of the ways that those models can be generalized is through the score function. So score function is the gradient of the logarithm of probability density function of ground truth data distribution. The good thing is we do not need to know this point explicitly. We only have to make sure that the synthetic data and the ground truth are matching. And the way to formalize it further was the stochastic differential equation approach, where we construct the diffusion process that is with the tractable SD, and we speed up the generation of the noisy data. So the pros is still we are getting the same high quality data samples, and we also can condition model on information not available during training. It does not mean that if we did not provide any combination of the samples on RAMs, it would work out. But we do not have to explicitly specify it to model to be able to use it at the later stage to make the conditional inference. So over here we can describe the noise process using the stochastical differential equation, where F is the drift of the data and G is the diffusion process, while the W is of W is infinitesimal Wiener process or Brownian motion. So overall, we can also write down the reverse SD and train the model to learn the score function that matches the data back to the original one. So here we got the sampling only that is marginally faster. And one of the interesting properties of this finding was the discovery of probability flow original differential equations. Those differential equations map the data to the same distribution as the stochastic differential equations. It means that first we can use them to make the noisy data from the meaningful distribution. We can map it to the Gaussian noise, and we can use the same technique to inference. This discovery led to the emergence of the consistency models, So it means that we can learn one deterministic model that can map the object from any trajectory point of the probability flow OD back to the original data distribution. So in theory, we can get the noisy sample and perform mapping back to the original point in one step. The authors originally approached the problem as the distillation process over the existing diffusion model, but they also discovered that the model can be trained uh, completely on standalone basis. And it still maintains the property that we can condition it on the information not available during the training. The consistency models was our choice for the uh, data generation process, and it consists. This architecture consists of UNet. It was implemented only for the image data, so we had to adjust it for tabular data, which we got for the gene expression in one of the key points here. We need to noise to create noisy versions of our data. In our case, we had only 4 distinct noisy points. And we are running it through the series of the attention blocks and the ResNet blocks. ResNet block modification was inspired by the TabNet, the 2019 model by Google, that used a simple transformer architecture to get the tractable tabular results. Another optimization for the model was the stochastic gradient descent weight averaging. Instead of using the very last update of the model, we're averaging the few last iterations of the SGD, and it results in more stable results, in more stable outcome. But to further speed up the learning process and the sampling, we moved our model to the Cerebrus wafer scale cluster. So you see this one chip that can run up to the 900,000 cores at once. But the challenge that we encountered was the middleware, MLAR, and the standard middleware that is used on majority of the AI accelerators, such as Grok and Sambanova. And one of the caveats of it was the requirement for the static computational graph. One of the something one of the issues with this was the inability to generate random numbers during the program execution. So we had to make the and to validate our results, we tried to run the unsupervised learning algorithm and see the split among the synthetic and ground truth data. So you can see for the majority of the outcomes, we are getting a very good mix of the ground truth and the synthetic data, but sometimes, and it means with the large number of clusters, the algorithm still can identify the synthetic data as separate cluster. I would like to thank our team and ILCF team that supported us with the computation, as well as the Cerebrus company that walked us through the process. Thank you, everyone. Any questions? Yes?
I would really love to talk to you later. But just for now, in my experience with tokens and the RNA-seq data are like, the fact that because it's all like cells, the correlation between them is very high, and thus models have a lot of trouble distinguishing between samples. How do you deal with that problem?
So yes, that's still a work in progress. So one of the approaches that we are trying is to use rejection sampling. So we use additional models to try to classify certain cell lines, and we basically filtering out everything that the model is uncertain about.